Hello everyone, this is Biochemistry 1.2 Cell Transport Mechanisms. So you know that the permeability across the cell membrane depends upon solubility in lipids, not on their molecular size. The water-soluble substances require a carrier. As the lipid membrane, is hydrophobic, so hydrophobic substances easily cross the membrane, whereas the hydrophilic substances require carrier molecules. Now, first we would like to highlight the classification of transport mechanism to you. The transport mechanism is classified into passive transport and active transport. The passive transport is further classified into four types, that is simple diffusion, osmosis, facilitated diffusion, and ion channels, which are specialized carrier systems. The active transport system is carried out by pump and vesicular transport mechanism that drive molecules against concentration gradient using energy. Now this is the whole schematic diagram. So you can see that the membrane transport is of two types, passive transport and active transport. Passive transport includes simple diffusion, osmosis, facilitated diffusion. Example is aquaporin. It also includes the fourth type, that is ion channel transport, which has three types, that is ligand gated channel, volt gated channel, and ionophores. Ionophores again are of two types, that is mobile ion carriers and channel formers. Now the second part, the active transport, is of three types chiefly, that is primary active transport, secondary active transport, and the vesicles. The mechanism of action of the active transport is by pumps and vesicles, and it is of the mechanism of is of two types, that is uniport and co-transport. Co-transport is again of two types, symport and antiport. So we have already discussed about the passive transport. We are now going to discuss the types, each type with their examples. So let us begin with passive transport. Passive transport, here the cell requires no ATP or no energy. The movement is from higher concentration to lower concentration. So this is known as transport based on concentration gradient. So the first type is simple diffusion. Simple diffusion is defined as random movement of particles from an area of higher concentration to an area of lower concentration. Now you can see this small animation. The second type of passive transport is osmosis. Now osmosis is diffusion of water through a selectively permeable membrane. Water moves from higher to lower concentration. Now let us see what are the different types of solutions that the cells here given below are in. Now this is the animal red blood cell. All the three are red blood cells, but they are in different types of solutions having different concentration of salt in it. So when the solution is hypertonic and the RBC is inside such a hypertonic solution, it will shrink because the water from the RBCs will come into the outside medium. In the isotonic solution, the environment or the amount of water molecules inside the cell and inside the solution are same, so the cells will maintain its actual structure. In the hypotonic solution, where the water molecules are more in the outside environment, So they will enter the RBCs, and the RBCs will swell and they may rupture, leading to hemolysis. In passive transport, facilitated diffusion of specific particles through transport protein is done in the membrane from higher concentration to lower concentration. So you can see here in this animation that this is simple diffusion where there is no carrier protein or no channel over here, and the molecules are just passing through diffusing through the membrane from higher concentration area to a lower concentration area. On the other hand, in facilitated diffusion, you can see that here there is a protein involved. There is no energy utilized, but a protein is assisting the movement of specific particles or ions from an area of high to low concentration. So the transport proteins here are specific. The transport of large molecules or charged molecules are done, and it is bidirectional and rapid. So compared to simple diffusion, facilitated diffusion is rapid because the transport protein is assisting the molecules to go from one side to the other. The mechanism is the ping pong mechanism. Now in the next slide we'll see what is the ping pong mechanism. So the ping pong mechanism includes the pong state, that is the active sites exposed to outer side of the membrane where the solute binds. Binding leads to change in the conformation of the protein, and in the pink state the active site face the interior. Uh, it releases the solute molecules into the cell and reverts the protein back to the pong state. And the examples of this are glucose and amino acid transport. So now we have simple versus facilitated diffusion, which I've once already explained, but for your clarity again, this is a lipid bilayer membrane, and this side shows lipids, which is a hydrophobic molecule easily crossing or diffusing across the membrane. And this is known as simple diffusion. Here the water molecule is unable to pass through the membrane because it is hydrophilic. So with the help of a protein, it will be able to go inside the cell. This is facilitated diffusion. And this structure is known as the protein channel. Now the example is of aquaporin, which are known as the water channels. They are known as the plumbing system of the cells. Water moves rapidly in and out of the cells. These water channels are tetrameric proteins that was discovered by Peter Agre and Roderick McKinnon. There are more than 10 aquaporins found in humans. They are numbered aquaporin 0 to 9 and further. Aquaglyceroporins transport small uncharged dissolved molecules such as glycerol, ammonia, carbon dioxide, and urea. This is the structure of aquaporin, which is also known as the water channel. So you can see that the steroid hormones, carbon dioxide and oxygen, they easily cross the membrane. They just diffuse across the membrane as per the concentration gradient. On the other hand, the water molecule is not able to pass through the membrane, so the presence of a water channel or a protein is required, which helps the water to move from area of higher concentration to lower concentration. The clinical application of aquaporins. There is significant presence of aquaporins in kidneys. The proximal tubules include aquaporin 1, 6, 7, and 8, whereas the collecting duct of the kidneys have aquaporin 2, 3, 4, 6, and 8 in large numbers. The aquaporin 2 reduced expression is seen in diabetes insipidus, where there is excessive urination volume. Now we come to the next example, that is ionophores. The ionophore is a lipid-soluble molecule synthesized by microorganisms to transport ions across lipid bilayer. These are of two types, that is mobile ion carrier valinomycin. It binds to a particular ion, shields its charge from the surrounding environment, and facilitates its crossing the hydrophobic lipid membrane. So this is how it enters the cell. So this is the mechanism that is seen in certain microorganisms. Now the second example is that of channel formers. An example is gramicidine. These are produced by microorganisms. They introduce a hydrophilic pore into the membrane, allowing the ions to pass. So you can see these are the ionophores. This is a channel former which forms pores into the membrane and allows the ions to pass from one side to the other across the lipid bilayer. And this is the mobile carrier where the ion charge is shielded and then it is transported to the other side. So these are the two channel formers. Now the next part is the ion channels. These ion channels are transmembrane proteins. They are selective transporter of one particular ion. For electrolytes like calcium, potassium, sodium, and chloride. Regulation is by voltage, ion, or they are mechanically gated processes. They are important for nerve impulse conduction, synaptic transmission, and secretion of biologically active substances. Now let us see the types in the diagrammatic form. Work. As you can see, the first one is the volt gated channel. Here there is voltage across the membrane, and as soon as there is change in the voltage across the membrane, the gate is opening and allowing the ion to pass. The second type is the ligand gated. These are again of two types, that is extracellular ligand and intracellular ligand. So in the extracellular ligand gated channel, a ligand or a molecule binds to its site at the receptor. Here it has bound to it, and after it binds to its own site, there is change in the conformation of the protein, which will lead to its opening, and once the gate is opened, the ions can pass through it. And the second one is the intracellular ligand, where the ligand is present inside the cell, and here it binds to its site, which leads to conformational change, and the gate will open and allow the ions to pass. So in extracellular ligand gated channel, the ligand is outside the cell, and in intracellular ligand gated channel, the ligand is inside the cell. Now let us see a little more in detail about volt gated channels. So as you can see, it is opened by membrane depolarization. It involves nerve impulse conduction, and common examples include sodium channels and potassium channels. You can see here, this is the sodium channel and this is the potassium channel, and once the voltage of these ions changes across the membrane, it will open the gate and the ion can pass through. So local anesthetics like procaine block these channels. Now the second type is the ligand gated channels. It is opened by binding of effector or ligand, acetylcholine which opens sodium channel and generates action potential in postsynaptic membrane. That is one example. The second example is of inositol triphosphate, which is also a ligand. It opens the calcium channel in sarcoplasmic reticulum, and amylogenin in the enamel acts on the calcium channel and opens it for the calcium to enter in. So these are the three examples of ligand gated channel. The ligands here are acetylcholine, inositol triphosphate, and amylogenin. This is a diagrammatic expression of ligand gated channel. You can see that this channel is closed here, and once the ligand binds to the channel, there is change in the conformation, and now the sodium ion can enter inside. Now we come to the channelopathies. That is, channelopathies are diseases caused by disturbed function of ion channel subunits or the proteins that regulate them. The diseases may either be congenital, mutations in the encoding genes of chloride, sodium, potassium, and calcium channels, or it may be acquired, often due to autoimmune attack of an ion channel. Channelopathies. Channelopathies are due to abnormalities in proteins forming ion pores or channels. These include some of the examples such as abnormality in chloride channel may cause a disease called cystic fibrosis. Abnormality in sodium channel may cause Liddle syndrome, and abnormality in potassium channel may cause periodic paralysis. The clinical application of channels. Local anesthetics like procaine act on sodium channels. Defect in sodium channel causes myotonia, which causes increased muscle excitability and contractility. Potassium channel mutation in the long QT syndrome leads to an inherited cardiac arrhythmia. The retina is excited by a photon by closing cation specific channels by hyperpolarization of rod cells. And in cystic fibrosis, there is defect in the CFTR gene causing a defect in the transport of chloride transporting channel. Thank you everyone for watching. Subscribe to my channel and enjoy learning biochemistry.